C3. Today our topic is the C3 cycle, and C3 cycle has the three phases. First phase is the carboxylation, and second phase is the reduction, and the third phase is the regeneration. In the first phase, in the carboxylation, the assimilation of carbon dioxide takes place, and in the reduction, there is the actual reduction of the 1,3 bis PGA, and in the regeneration phase, whatever the RUBP ribulose bisphosphate is utilized in the carboxylation is formed in the regeneration phase. And now we are going to start one by one. And first, when the carbon dioxide is absorbed from the stomata, the carbon dioxide and the RUBP are combinedly form the ketoic acid. Already RUBP is a 5C compound, and again one carbon dioxide is added to this 5C compound to form 6C compound. It is a ketoic acid. This ketoic acid is highly unstable, so it is break down into the 3C compound, so that it is the phosphoglyceraldehyde, and this phosphoglyceraldehyde undergoes the phosphorylation reaction. Wherever the phosphorylation reaction takes place, there should be a addition of phosphate. Here addition of phosphate is by the ATP. The ATP gives one phosphate to 1 PGA, and ultimately there is 12 PGAs. So 12 ATPs gives the 12 phosphates for the phosphoglyceraldehydes, and it results to form as 1,3 bis PGA. Here wherever the phosphorylation reaction takes place, and there is the ATP, wherever ATP it is present, there will be the enzyme kinase. Here it is a phosphoglyceraldehyde, so phosphoglycerokinase enzyme is utilized here to form as 1,3 bis PGA, and this 1,3 bis PGA undergoes the reduction and dephosphorylation. In the reduction phase, there is the addition of hydrogen. The addition of hydrogen is done by the NADPH. NADPH gives hydrogen to the 1,3 bis PGA. And next, dephosphorylation. Dephosphorylation is done in the form of H2PO4 minus, where the phosphate is removed from the 1,3 bis PGA. 1,3 bis PGA in presence of G3P dehydrogenase, it is converted into G3P. There are the 12 G3Ps, so it is called as the G3P pool. Large number it is present, so it is a G3P pool. And next we have to arrange this for the easy understanding. And here the G3P one, every pair of G3P, one pair, 6 pairs of G3P, 12 G3Ps are there, 6 pairs, and each G3P is of 3C carbon, 3 carbons. Triose, G3P is a triose, and 1,3 bis PG is a triose, and PGA is a triose. So all these trioses undergoes in the reaction. If one pair of the G3P goes into the cytoplasm, where actual glucose formation takes place. Formation of glucose. And what is the use of C3 cycle? మనకు photosynthesis టైంలో గ్లూకోజ్ ప్రిపరేషన్ జరగాలి. సో మెయిన్ ఎయిమ్ ఇస్ గ్లూకోజ్ ప్రిపరేషన్. One pair of G3P goes into the cytoplasm, and one of the G3P is converted into DHAP, dihydroxyacetophenone. Dihydroxyacetophenone is a isomer of G3P, and these two undergoes condensation. Both together form fructose 1,6 bisphosphate in presence of aldolase enzyme. Wherever the two different substrates combine to form a single compound, this type of reactions are called as a condensation reactions. Wherever the condensation reaction takes place, there is the aldolase enzyme. And here the fructose 1,6 bisphosphate is converted into fructose 6-phosphate or fructose monophosphate. Here the phosphate is released from the fructose bisphosphate, so it is called as the dephosphorylation reaction. It is takes place in presence of phosphatokinase. It is also called as fructose phosphatase. And next one is fructose 6-phosphate is converted into the fructose. Here again this phosphate is removed, so that's why it is a dephosphorylation. Wherever the dephosphorylation is there, there is a phosphatase enzyme. It is a fructose phosphatase, and we got the fructose and this fructose is converted into the glucose. Fructose and glucose are the 6 compounds, 6 carbon compounds. These are the hexoses, which are the isomers. So it is involved in the isomerization reaction. Here the hexose isomerase enzyme is involved, and now ultimately we got the glucose in the cytoplasm. And our next step is to regeneration of ribulose monophosphate regeneration is our next aim. So how it is formed? One of the G3P out of two, one of the G3P pair is converted into DHAP. G3P and DHAP are the isomers. Here this reaction is called isomerization reaction. Here the triose isomerase enzyme. These two are 3C compounds, so these are called as trioses, and this is involved the triose isomerase enzyme. Triose isomerase enzyme. It is a DHAP, and DHAP and G3P both are together forming a single compound as they here. So both two different compounds resulting in a single compound. It is a condensation reaction. Everywhere, wherever the condensation reaction, there is aldolase enzyme. Here in presence of aldolase enzyme, this DHAP and G3P is converted into the fructose 1,6 bisphosphate, and this fructose 1,6 bisphosphate is converted into fructose 6-phosphate. It means that there is the removal of phosphate. It is called as the dephosphorylation reaction. Here the enzyme is the phosphatase, fructose phosphatase, and this fructose phosphate, fructose monophosphate, and glycerolate 3-phosphate both together to form two different compounds. రెండు కలిసిపోయి రెండు డిఫరెంట్ కాంపౌండ్స్ ఇస్తున్నాయి కాబట్టి దీన్ని కంబైనింగ్ రియాక్షన్ అంటాం. ఇక్కడ ఏమైంది, రెండు కంబైన్ అయిపోయి కొత్త కాంపౌండ్ ఇచ్చింది కాబట్టి కండెన్సేషన్ రియాక్షన్. రెండు డిఫరెంట్ కాంపౌండ్స్ని ఇస్తే దాన్ని కంబైనింగ్ రియాక్షన్. వేరెవర్ ద కంబైనింగ్ రియాక్షన్ ఇస్ there is a trans-ketolase enzyme. In presence of trans-ketolase, G3P and fructose 6-phosphate are converted into the erythrose 4-phosphate and xylose 5-phosphate. Erythrose is a 4C compound, it is a tetrose, and the xylose 5-phosphate is a 5C compound, it is a pentose. Xylose and ribose are the 5 pentose compounds, those are the isomers. Remember this. And G3P is one of the G3P is converted into DHAP, which is the dihydroxyacetophenone. G3P and DHAP are the isomers. So here is the isomerization reaction, and the enzyme is triose isomerase. And this DHAP along with the erythrose 4-phosphate, two different compounds are resulting in a single compound. It is called as a condensation reaction. Wherever the condensation, there is aldolase enzyme. In presence of aldolase, it is a 4C compound and it is a 3C compound resulting into the 7C compound, 7 carbon compound. 7 carbon means pseudoheptylose. Hepto means 7, so pseudoheptylose 1,7 bisphosphate. Here 1 phosphate, here 1 phosphate. So 2 phosphates. Indirectly it is called as a bisphosphate. 1,7 bisphosphate. 1,7 means first carbon has a phosphate and 7th carbon has a phosphate. And the next step is the dephosphorylation, where pseudoheptylose 1,7 bisphosphate is converted into the pseudoheptylose 7-phosphate. Wherever the dephosphorylation reaction takes place, it is the phosphatase enzyme. So pseudoheptylose bisphosphatase enzyme is acting here to form as a pseudoheptylose 7-phosphate, and the pseudoheptylose 7-phosphate reacts with the G3P, which is a 3C compound, and it is a 7C compound together to form the actually it forms 10C compound, but it is not stable, so it results into the two 5C compounds, ribulose 5-phosphate and xylulose 5-phosphate. Two different compounds are combining to form two different compounds, so it is a combining reactions. Ribulose 5-phosphate and xylose 5-phosphate. Ribulose 5-phosphate, it is also called as ribulose monophosphate 1-phosphate is present. Here the ribulose 5-phosphate isomerase enzyme takes place, isomerization reaction, and this xylose 5-phosphate. Here there are the two xylose 5-phosphates, and here there are the two xylose 5-phosphates, and these 2 to 4 xylose 5-phosphates are conversion. Means it is both xylose 5-phosphates are converted into ribulose monophosphate. These two are the isomers or epimers. So here the RUMP, ribulose monophosphate epimerase enzyme is involved, and these two 4. So 4 RUMP of 5C compounds are formed. This is of 2. This 2 and this 4 results in the 6 of 5C compounds. Ribulose monophosphates. It is a pentose. These 6 ribulose monophosphates are, again we got ribulose monophosphates are converted into ribulose bisphosphate. It means phosphorylation reaction. Phosphorylation, addition of phosphate takes place by the ATP. ATP gives 1 phosphate to the 1 RUMP. 6 ATPs gives 6 ADPs and 6 ribulose bisphosphates. And here the cycle is one part of this, and another one is the how many ATPs are utilized. Here the utilization in the reduction phase, there is the utilization of 12 ATPs. 12 ATPs are used in the reduction phase, and again here the 6 ATPs are used in the regeneration phase. 6 ATPs are used in the regeneration phase, and 12 NADPHs are used in the reduction phase. We have to calculate for 1 carbon. 6 carbon dioxides require 18 ATP. Count down, here 12 and here 6. Totally 18. 6 carbon dioxides requires 18, and 6 carbon dioxide require 12 NADPHs. So 1 carbon dioxide requires 3 ATPs, and 1 carbon dioxide requires 2 NADPHs. And this calculation part is very important for the NEET. Almost the question comes from here in the C3 cycle. Please remember this for the problems. Thank you.